We have a second session this morning where we will be able to touch a little bit more on aspects that belong to the discussion around a healthy diet that actually we haven't heard about a lot. We all know we don't eat nutrients, we eat foods. And I think for each of the food solutions that we try to develop, it's important to ask ourselves the question is what is the position of these food products in the total diet? In this session, we will have three eminent speakers that will touch upon different elements that will help us to have this discussion around a healthy diet. The first speaker, I will introduce her in a minute, will pay much more attention to energy density of foods and how insights into that can help us to actually teach us what is the position of certain foods in the diet, and especially if we think about some aspects that already surfaced yesterday, which is what are actually the nutrient needs of different parts of the population, and they might be very different if we talk about young kids or pregnant women than if we talk about adults. And that is aspects that she will go into. Then, of course, it's really important that in this environment, it's in the end the consumer that makes choices on what belongs in a healthy diet. It's not only the consumer itself, it's the consumer's knowledge that will be an important determinant, but also the environment in which the consumer lives. Cultural beliefs, social background, but also the food systems that this person is part of. And our second speaker will touch upon that. And then last but not least, we have a contribution that will take us deeper into personalized diets. Because what we consider to be a healthy diet may differ between individuals. And the science is advancing there and giving us lots of clues on how we can determine better what are people's personalized nutritional needs. So with this short introduction, I would like to start with our speakers, and I would first like to introduce Professor Barbara Rolls. Due to travel difficulties, she will join us virtually, but she will not be here in the room. Professor Barbara Rolls is currently professor in nutrition sciences at the Penn State University in the USA. So with that said, Barbara, the floor is yours.
So we've been hearing a lot about nutrient-rich foods and fortification and how to improve foods. I want to talk a bit about how do we get people to actually eat those foods. And I know the audience is very diverse in their interests. Our interest has been primarily in overconsumption and obesity. So just with that little bit of introduction, let's get started. So I think by now we know that there are a lot of data that indicate that big portions lead to overeating. There have been a number of different studies with different types of foods in lots of different countries. And most of them started with amorphous shaped foods, those that are hard to judge in portion size. But the portion size effect extends to unit foods, sandwiches, pieces of pizza, beverages, snack foods, even vegetables and fruits that are low in energy density. And that'll be a big focus of what I'm going to talk about. How do we get people to eat more of those? The studies have been done both in labs and naturalistic settings. Most of the studies that have been done are single meal or a couple of meals. They're short term. So this is a study that we did in adults, trying to see if the effect persisted, because when we would first start talking about this, my colleagues would say, okay, fine, you overeat at one meal, you're going to compensate at the next meal. So in this study, we were able to feed people all of their foods over a period of 11 days with their usual portions that are plenty big enough so that they're not just cleaning the plate. And then across all the meals and snacks, we increased the portions by 50%. And here's what we found. The men are at the top, and over these 11 days, they accumulated 4,600 extra calories. The women over 5,000 extra calories. There was no sign that they were compensating over this 11-day period. So the effect is robust, persistent, and fairly large. So From there, we wanted to look at preschool children, young kids, 3 to 5-year-olds, because they're in a formative stage where their eating habits are developing. Plus, it's thought that they have an innate ability to self-regulate their food intake. So they should, in theory, be able to resist the effects of portion size, energy density, et cetera, on their intake and self-regulate. So here's what we did in these 3 to 5-year-olds. We used a crossover design, and it was on portion size. The most you can do, we're testing them in their child care centers. So the 5 weekdays is pretty much as long as we could go because we're providing all the food. And here you can see the typical amounts of food that we served over a single day. Again, we have the usual portions, and all the foods we feed have to fit all the school guidelines. So these are pretty healthy foods. And then we had a condition where we increased portion size, again, by 50%. The menus differed each of the 5 days. And we had a monitor who was eating with them. So here's just how we did these studies. I'm going through this because I'm going to show you 3 different 5-day studies in these children. So we prepped all the food in our lab. We had to carry it to various child care centers around the community. We plated up, weighed the food. The kids wore accelerometers around their waists. They ate in their usual setting, and we carefully weighed back all of the food. So we have measured intake over these entire 5-day periods. And here's what happened when we increased the portion size by 50%. This is, again, cumulative intake. And here you have the usual portions and the bigger portions. And this indicates that the 2 lines started significantly diverging right at lunch on the first day. And again, there was no sign of a compensatory response. And they ended up overeating by almost 800 calories over those 5 days. The story gets even more interesting. In these kinds of studies, we're always trying to look at individual differences. Who is the most susceptible to these food cues? And of course, body weight, body size has always been a primary target. So here we're looking by BMI percentile at the daily energy intake as a percent of energy needs. Since they had the accelerometers on, we could do that. And you can see down at the lower BMI percentiles, there's only a small response to the portion size effect. As the children get heavier, and this is not a very heavy group of kids, relatively speaking, we got a much more significantly different effect. So here we are. Large portions start affecting intake at an early age, and it's affected by weight status. The effect is largest for energy or calorie-dense foods and those that are most liked. And the effect is persistent. And then many people are just not aware of how much they should be eating. What is the right size of a portion? How much should people be eating? And how do we prescribe that when we differ so much in size, physical activity, et cetera? Well, this has been a problem that's been discussed, and many different solutions have been proposed. We have talked about economic environment, pricing, taxation, subsidies, political solutions requiring formulation standards, standard package sizes, physical environment changes, decreasing portions of high energy dense foods, larger portions of low energy dense foods. And I'm going to focus on that approach, plus under personal community environment. Obviously, we can improve nutrition knowledge. I'm also going to talk about tools such as My Plate to guide choices. I don't have time to talk about all of these different possibilities, so let's dig in on proportionality, since that is being recommended in a lot of different countries now. My Plate in the US emphasizes that half your diet should be fruits and vegetables. Other countries are proposing different proportions of fruits and vegetables, but in general, proportionality has become quite a popular tool for dietary guidance in recent years. So the interesting thing about this is there's almost no data indicating whether it actually works. We did a study in adults some years ago, before this was really a popular approach, and we just did a single lunch meal, showed that substituting vegetables for other parts of the meal increased vegetable intake while lowering total meal energy intake. And here you needed to keep the energy density of the vegetables low, no butter sauces, cream sauces, and the palatability high. If you want to increase vegetable intake compared to other foods, they have to be relatively palatable. This kind of approach has also been extended in the Netherlands to restaurants. And this group has done several studies now where they've gone into restaurants, other types of cafeterias, buffets, and shifted the proportion of vegetables and meat. So they've increased the vegetables by doubling them, and then slight reductions in the meat or fish. And they also had some unmanipulated vegetable side dishes. And you can see they, by doing this, increased vegetable intake and decreased the meat and fish. And the customers were satisfied after these meals. So that's great. These are single meal studies still. What happens over a longer period of time with shifting the proportions of food on the plate? And here again, this is one of our 5-day studies in the preschool kids. So here we have again the control condition, which is typical amounts they would eat of the prescribed foods. And then we wanted to compare what happens if you just add more fruits and vegetables as side dishes or snacks, or you substitute the fruits and vegetables for other meal components. And we wanted to compare addition and substitution because the guidelines usually emphasize substitution. So do they both work in terms of increasing fruit and vegetable intake? But we didn't find any differences across the 5 days. So the effects were persistent. So here we're giving averages across the 5 days of vegetable and fruit intake. And here's the control condition. And you can see that addition and substitution increased intake of both the vegetables and fruits. Not surprisingly, the kids eat more fruits than vegetables. So there were persistent effects, and we did get increases, particularly in the vegetables. What about energy intake? Did we get any effects there? And you can see that by adding vegetables to the meal, the kids didn't eat less of the other foods, so they actually ended up eating more calories. With the substitution, we got a significant decrease in energy intake. So where are we with this? How should diets be reportioned? First, want to emphasize there are a lot of different ways to increase the proportion of vegetables and fruit in the diet. In the study I just talked about, we manipulated side dishes and snacks. You can also do a first course. We've done a vegetable soup or carrot sticks at the beginning, and bigger portions increase intake in kids. So doing it when they're hungry. You can modify recipes. We actually find that tucking vegetables into recipes, even covertly, is a really effective way to get kids to eat more vegetables. And obviously, adding some variety also works. So when we're increasing the portions to boost intake of these nutrient-rich foods, one of the key messages is to keep their energy density low while ensuring they're appealing. And then if we want to manage intake, substitute, not just add more lower energy dense options for the higher energy dense options. So I've been talking a lot about energy density. So what happens when you vary the energy density over 5 days in these preschool kids? Will they show a compensatory response? So here we have our baseline energy density. When you're looking at effects of energy density, you don't want to vary the whole diet. You need to give them some unmanipulated foods so that they can compensate and eat more or less if they're responding to the change in energy density, which here was we manipulated the energy density of the 3 main dishes at meals and at one snack. And we either lowered the energy density of those 4 items by 20% or increased it. So what did these kids do? Here is their cumulative intake again. The orange line is the baseline. And you can see again, there's no sign of a compensatory response over the 5 days. The children, when they had the higher energy dense meal components, overate by around 84 calories. And when they had the lower energy dense items, they underate. So what about weight status? Here again, we're finding that BMI does influence the response to energy density. So the kids with the lower BMI percentile they didn't respond much to the variation in energy density. They're slightly under eating. As the children get heavier, they're showing a bigger response. So that with the high energy density items, they're overeating by 104 compared to control, or undereating by 141 kcals per day. Okay, so we've shown that typical variations in portion size and energy density lead to sustained changes in kids' energy intake and in adults as well. The children didn't compensate for the overconsumption related to the increases in portion size over these 5 days, and the heavier children were more responsive to the variations in both portion size and energy density. So what happens if both portion size and energy density are changing together? Have we topped out on how much is going to be consumed, say, when we give big portions? So we conducted a study where we looked at the combined effects of changing portion size and energy density. So we had 3 different portions and 2 different energy densities. And these are very common, popular foods. This was not covert. This was just the foods that kids would typically eat. So, for example, we have grilled chicken versus breaded chicken, macaroni and cheese with more fat to make it higher energy density, a starchy vegetable compared to broccoli, sweetened versus unsweetened applesauce, higher fat milk. And here, just to show you the difference between the lowest energy density, smallest portion, and the biggest portion, highest energy density, this meal has 455 calories. This almost 1,300. I think a lot of people wouldn't necessarily notice that the difference is this big. This is a single meal now. Let's see what the kids did. So you can see that with the higher energy dense meals, the kids are in general eating more than they are with the lower energy dense. And we have a systematic portion size effect in both types of meals. So that comparing the smallest lower energy dense to the biggest high energy dense, we had 175 kcal difference, or a 79% difference. These are little 3 to 5 year old kids, so that's a big effect. These kinds of ideas are only just getting out into the real world. And I wanted to show you this study from the UK that was published recently. This was conducted in 19 worksite cafeterias of a large grocery chain, with the consumers were primarily manual workers. And so what they did in this study is they, first of all, went in and they substituted lower energy dense items, lower calorie items for some that are higher in calorie. They couldn't do it with all the foods. Obviously, there's a lot of practical issues when you go into these kinds of work sites. And after a few weeks of that, they added in also some decreases in portion size of main dishes, sodas, some of the snacks, desserts. And you can see this was a 25-week study. Between sites, there was a lot of variability. There was some difficulties in compliance with some of the sites. But overall, here's what they found, that just shifting the availability, they got around a 5% decrease. If they added a decrease in portion size, they got overall an 11.5% decrease in the amounts of calories purchased, or the change in energy purchase. They couldn't look at this at the individual level. So this is showing that you can, to some extent, environmentally engineer, although with difficulty, and reduce energy intake. All right, so in summary then, what we have is that portion size does not have to be a bad thing. I think it's been vilified in terms of obesity. We can use it strategically through increasing the proportion of the vegetables, fruits, other lower energy dense, nutrient-rich foods. And you can see from this example, this is the same number of calories. This is just shifting the proportions. So people actually get much bigger portions if they're shifting towards lower energy dense options and end up more satisfied. So if you want to moderate energy intake, you should substitute lower energy dense options for those higher in energy density. Serving ample portions of lower energy dense, nutrient-rich foods when hungry works well, incorporating into recipes, as I said. And through all this, we have to emphasize palatability. If you want to increase, for example, vegetable intake, they have to be relatively palatable compared to the other foods. And if we apply these strategies, people can have satisfying portions, they can have a full plate, and they can improve their diet quality. So thank you very much.
Thank you, Barbara, for a very, very clear presentation that really emphasizes how much attention we need to pay to low energy dense, but nutrient dense foods and the position of those in the total diet. So, like I said, this session will pay attention to different aspects, and now we move to our second speaker of the session, which is Professor Monique Raats, currently Director of the Food, Consumer Behavior and Health Research Center of the University of Surrey. So I'm really looking forward to Monique's contribution to this session. Monique, please.
Thank you very much for the opportunity to speak here, and it's really nice to be part of such a broad discussion and think about the individual in that context. So I'm going to start with really thinking about what does food mean, and this is a very nice review that was done taking learnings from sociology and anthropology, and what does food mean for us all, and this model sort of separates out three things. So consumption, that's the way we talked a lot now in the meeting about what it is we do with food, and so the hedonic side, do we like it, but also in some cases the symbolic side. We can use food to symbolize power. Organizations that serve champagne are saying something about the sort of organization that they are. But we also use food to transfer things, emotions, culture. I had a very personal experience last week where food was really, I have someone dealing with severe mental health issues and making communication very difficult. And we were able to use cooking and spending time in the kitchen together as a means of having conversations after long periods of not having been able to do that. So food is really a signifier, and we use that in our houses. I think COVID has showed us how important the kitchen and the spaces that we prepare food are, and if they're not there, what that means. Culture is also something that we transfer through food. We saw a lot of that happen in the last few days. It says something about us as countries, as people, and we also own dishes in, I say that in quotes, in certain ways because they belong to particular cultures, and they are very important in that. And then lastly, identity. So they say something about us as individuals. We could also introduce in there as groups. And we use foods to communicate things to do with nationality. And nationality could be the boundaries of countries, but it could also be regions. We see quite often commonality in parts of the world that have very similar diets. And sometimes that could possibly be used to also bring people together. So we saw this in Barbara's presentation, so one way of thinking of all the levels of influence on an individual, and she did that very elegantly in the space of thinking about portion size as a way to think about managing food choices. But we have all the levels: the social environment, the community environment, the organizational, and the public policy environment. And it's usually different scientists who get involved in doing research to take account and better understand how each of these levels functions. And sometimes I'm a little bit uncomfortable in rooms where we talk about the science, and then what we're talking about is the biological, the medical, and maybe the behavioral sciences when we're thinking about. But there's a whole host of other sciences that really help us understand and answer some of the questions and bring together. And the really translational sciences, when can we do that adequately? So the framework for thinking about food systems is very complex, and we see here the consumer is depicted in the green box. Quite often, we have to sort of think, is it just consumer behavior? How does that relate to all the other parts of the system? And how much do other parts of the system really consider? Or is it an afterthought, perhaps, that we sort of say, we do our business, and then we speak to and we ask and we check, is what we've done acceptable? Here's an example of a nice review that was done about thinking about food trends. So we have a number of trends that we've been speaking about in the meeting here. And we might want to think, what do consumers make of these? When do we start talking to them? Is it once we've come up with the concept, once we've done quite a lot of science? Or might we think about doing something where we really bring the public into the conversation much, much earlier and make them part of the conversation around how we do science, what questions we address? And this concept is what we call research and innovation with and for society. The phrase that's quite often used is responsible research and innovation. And it's a framework for thinking about how does science and technology fit in society, and how can we use processes to ensure that we're doing that adequately in the science that we're doing. And so it's really about how do we bring together the different actors, and here we've sort of grouped them into four: the public sector, government, governance, citizens, the third sector, organizations, civil society organizations, again, quite a broad collective group, industry, and the research and innovation community. And each of these sectors can have ties, and people can have multiple alliances between these. And what I haven't really depicted here is the role of the media, who are sort of a glue between many of these sectors, and they're one that you might also want to consider. So this is the definition. It's about having a transparent, interactive process by which societal actors and innovators become mutually responsive to each other with a view to ethical, acceptable, sustainable, and societal desirable solutions, and having a good innovation process. Thank you. And so we have processes that we can sort of bring into projects, and my team quite often do that in projects. We're sort of having studies that help facilitate those discussions, and what many people are used to in projects is to sort of think about, well, we'll do that at the end. We'll communicate and we'll disseminate once we've done our science. And what we're trying to do is really push bringing in that conversation much earlier. And I think when people start to take part in those processes, are quite surprised sometimes about some of the thinking that can be brought in. And it's really become very much part of some funding streams now, particularly in the area of health research, that you have to actually have spoken to your target audience before you start to come up with solutions and show that you've really considered that in the doing of your research. So we're starting to really build that, and it's expected that you build that into particularly large programs of work. Another way and that's sometimes brought out now is to use the term citizen science, and this could be research that involves volunteer, non-professional, people who might not have been as trained in science in the doing of research. And this review is a quite nice one that sort of shows where has that all been brought in. And again, we see examples from across the whole food system in which that's been done, particularly in agriculture. I think there's quite some work where people have been brought in to help collect the data. And that's what I want to sort of highlight is when we do science, we have many steps: the setting of research question, the developing of how we're going to do the science, the collecting the data, the analysing and the sharing of results. And what we see from a review that was done in the area of health equity research is that we typically see that most studies really mainly involve the public or citizens in collecting data, and less typically in the phases like defining the research problem. And so we might want to sort of think, yeah, would we feel comfortable as scientists bringing people in very early on to discuss with them what it is we're thinking of doing and what they make of that? And so the idea, again, would be that if one does these things, again, there will be more openness, more trust and transparency in the take-up and use of science. And I think what we've seen in recent years is that we do have parts of the world that are, and parts of society that are very skeptical of research and its role that it plays in making decisions. This is, again, some work that was done sort of reviewing a lot of the big food systems reports that have been done. So again, it uses the same thinking shape. And if we look, the middle column is the reports that really consider consumptive behavior. So what we see in the food systems thinking is typically a lot of the work has been really the emphasis in the production end. And we haven't started to really incorporate, and I've got 2 slides here, as much consumptive behavior, and then even more interestingly, thinking of the outcomes of thinking about the food systems, the health research. So what we don't have is as much thinking having been done to think about how do we link consumption to health outcomes and then back into the system of how do we produce and create the food supply that we need. So my team has not just done the elegant work such as that Barbara just talked about, the experimental work that you can do on many topics, but we also do work which is around how do people think and talk about food. We've done historically quite a lot of work in the labelling space, front of pack labelling, and this was a study that we did a little way into the period we worked on front of pack labelling, and we went to the public themselves and asked them, can you please sort all these labelling schemes that we have and see what you make of them? And this is the typology the public came up with. And I think it's a very elegant solution to sort of show what the differences are between the different labelling schemes. And up until now, in the technical community, we were talking about things like interpretive labeling, describing the differences from a technical perspective. And what this really does is show that we have some systems that are directive. They tell the public what to do, the logos. We have some that we call semi-directive. There is some suggestion as to what to do with that. And then we have what we call the non-directive systems, the one that are basically like the back-of-pack nutrition information, where you need to then do quite a lot of decision-making how to make something of that. And so for us, that really, we took that then into the design of further studies to contrast and feed into the debate around, and we still have quite a long way to go to come to decisions as to how we're going to be doing that in different nations. We recently used the same method to sort of make sense of the world of sweeteners and sweetness enhancers, where we again gave the consumers a pile of cards saying, please sort these in any way that you want, to try and see how do they make sense of these terms that they might come across on food packaging. And what was really interesting here, the team we worked with, we did this in the UK and Spain, and I worked with some colleagues who don't typically do social science research. They were nutrition background, and they were a bit skeptical about, ooh, this is a bit strange doing this kind of thing. So we did it online because it was during COVID. And the experience they had as researchers, as part of the team, was really transformational. They started to then see, oh, this really it helps me make sense of some of the trial data that I'm doing, because we do many of these studies with the public in the context of clinical trials, where we have the opportunity to gather extra data. And here, the typology that came out was the distinction between artificial sweeteners and sweeteners from plant sources, the level of familiarity, and also the complexity of names. And so there we can then straight away make assumptions about how favourable these sorts of substances are going to be when people are communicated about them. So again, doing rigorous work around how do people understand the concepts helps us inform how to do further research. We also did it in the area of processed foods. We have a PhD student, Christina Sadler, who's been looking at how processed foods are thought about and classified. We did a review and looked at the different classification systems used across the patch, and we see many different systems. There isn't one definition. And what we tried to do here was also come up with a classification system to sort of show how are processed foods being classified. And basically there are 4 ways that one can do that: the extent of change, nature of change, place of processing, and purpose of processing. Those are the underlying concepts in the many schemes that exist and are being used for different purposes by different people. And then we see 2 of those, the place of processing and the purpose of processing, are ones that really relate to consumers in terms of they are really getting at the social-cultural aspects of, in terms of when we think about place, and the purpose of processing, that also really links to consumer behavior aspects. So again, the importance of thinking about the consumer in the context of how we as experts think and talk about food. We also did a qualitative study with experts to sort of see how do they think that the public thinks about food. So we don't always just do work with the public, we also sort of speak to experts and try and think, what are the underpinning ways, what are the assumptions you're making about human behavior when you think about what to do next. And here we were able to really highlight what are the major challenges associated with communicating about processed foods, that there are very broad concepts that need probably more detailed differentiation. There are disagreements about the scope and degree, and I think we've seen that, and I think there's quite some scope if we want to think about the role that these foods play to actually be able to attribute benefit, and how do we communicate benefit associated with processed foods. So yeah, being able to frame both the risks but also the benefits, and then there are then associated with that challenges for risk communication, and we have quite some knowledge about how to go about doing that we've learned in other areas, and I think we can take those to this space and really think systematically about each of the types of processing that we have. Then a little quick next step into the science. Psychology is one of the fields where we've drawn a lot of methodological knowledge from, and this is an overview of how we sort of can think about the area and that type of data that exists. It was a nice rapid evidence review done for the Food Standards Agency in the UK, and it really sort of separates out thinking that where people think deliberately about things, you're very conscious about what you do, and certain interventions typically would be associated with that. We have unconscious, much of what we do is very habitual. Most of us probably eat more or less the same meals on a weekly, perhaps monthly basis. Most of us maybe don't have a repertoire of more than maybe 10 meals that we typically eat. So to sort of treat food choices as everyday, a new thing is not perhaps how we need to sort of think about food, and it's also influenced by things like impulse, mood. And then the indirect effects, the environment also have quite, so all of these things interact with each other, and the models and studies, it's very important to sort of think what are the assumptions you're taking when you're going to think about how to then influence food choices. And here we can sort of see an overview of what strategies might promote positive food choices. So if we're thinking about deliberate, that might be supporting self-regulation, helping people set goals, monitor themselves, where you're very aware of what you do. But non-conscious ones, behaviors you might really target with things like the architect of the food environment, things that Barbara talked about. Food labeling fits in that space as well. But then if we go to the indirect, we start to sort of think of things like the social modeling of positive food choices, so the environment and how that influences. But what we see is that some of these processes really work for people who are very motivated. And if there are groups who aren't motivated, we might have to think of very different strategies. So again, thinking about who is it that we're trying to work with will really influence how do we select our choices. Almost there. So there are limitations with regards to consumer science, and as a field we've been really thinking about that a lot. I just have a few slides to really want to highlight a project that we've started about a year ago, led by a team in Wageningen, and it's called the Focus Consortium, where we're really trying to build community and be critical of the methods that we use, because typically we've really been coming from a very individualistic paradigm, not maybe needing to do a lot more work about the methods that we use. And it really ties in well with the last presentation in the previous session, that we're also trying to set ourselves up for being able to take and pool together data and be able to use techniques such as AI, machine learning, to look across studies and build bigger data sets and data pools. So we're trying to transform the discipline, make it more usable, integrate it with our other disciplines, widen the number of theoretical lenses, and really also broaden and be more critical. We tend to, as scientists, work with who we can get. It's sometimes very hard to bring people into studies. We don't sometimes want to think how many studies have been done with very easy populations, and if science is going to be credible, it needs to be done with populations where it needs to work. And I want to use the term that we have an overemphasis on weird data. That's data coming from a Western, educated, industrialized, rich, and democratic context, and much of the world isn't that. And so we really need to increase the amount of data that we have coming from populations so that we can make decisions that are adequate based on data that really fits those populations. So we have a long way to go as a science community to work to do that and have techniques that work in all of those contexts. So the mission here is to start to bring together. We are offering early career researchers to come into this community to link their data and become part of it. So I hope those of you that do behavioral data would consider linking up with our project. And we also have funds to have people come into our labs and make use of some of the research infrastructure that we have to do this sort of research around Europe. But it's really open to scientists from other parts of the world as well. And hopefully we will increase that network to much wider than just Europe, but we've had European funding to do this. So coming back to identifying responsible food innovations, we need to sort of think, what's the perspective? Are we taking a rational perspective? Are we thinking that behavior is irrational? Or do we want to build on social learning models? And if we're going to do that last thing, it means we maybe need to think of not consumers, but citizens, and think of this concept of food citizenship. And food citizenship means that we have responsibility as well, us as individuals, but us also as groups. And that really starts to change how we think about what we need to do, what the right solutions are. And that's what I sort of want to leave you with. I have a group called Consumer Behavior Group. I would like to transform that into a food citizenship and really starting to think about how do we, at an individual level, but also group level, start to transform food systems and empower people to be part of making the changes that need to happen. Thank you.
So our last speaker of this session is Professor Eran Segal from the Weizmann Institute of Science in Israel. His background is in computer science and applied mathematics, but I think we've already seen in the previous session how that type of knowledge could really help us to advance our thinking on healthy diets. Eran, please, the floor is yours.
Okay, thank you for the introduction and kind invitation to speak here today. Do we have the presentation? So I'm going to try and cover over a decade of research that we've been doing in this area. So I'll try to give an overview. So sorry, apologies if I go fast. So we started looking at the problem of nutrition, and as a healthy marker, we decided to focus on postprandial blood glucose response to food, so the glucose response in the 2 hours after you eat a meal, because that's very key to weight management. It's very key to disease like diabetes, cardiovascular disease, even cancer and other diseases. And it's also something that with continuous glucose monitors, we can actually measure in people and get a response to every single food that people eat. And based on this premise, we started the Personalized Nutrition Project in Israel about over a decade ago, where we recruited 1,000 people, connected them to continuous glucose monitors, CGMs, had them log all the food that they eat. So we had blood glucose response to 50,000 meals. And together with that, collected a very extensive profile of personal information about people, including blood tests, medical background, and microbiome data. And the study, which we published in Cell in 2015, showed that people have very different blood glucose responses when they eat exactly the same meal, as you can see exemplified here in the case of bread. We were then able to develop a machine learning algorithm that takes personal information, including microbiome data, and quite accurately predicts personalized blood glucose responses to meals, and show in a small intervention study, a short-term also one, that you can take people who are pre-diabetic and normally have major spikes in glucose levels, and you can tailor a diet to them with exactly the same amount of calories that sometimes even contains surprising foods like pizza and chocolate and ice cream, yet you can still fully balance their blood sugar levels. This is a trace of glucose levels over a week. And we recently completed a proper randomized clinical trial in 200 people with prediabetes, which we randomized into either our standard of care Mediterranean diet or our algorithm-based diet, and looked at the changes in glucose levels, measured either through hemoglobin A1C or averaged through a CGM that we put on people for half a year straight. And the study had 1 month of profiling, then 6 months of intervention, and another 6 months of follow-up. We collected a lot of clinical information, metabolic information, fat enzymes, and so on, lipids on people, also some exploratory microbiome and metabolomic analysis. And here's an example of one participant on the algorithm diet, where this is a trace of CGM data for a full month, and you can clearly see spikes in glucose levels indicating that this participant is indeed pre-diabetic. And the same participant, once we start him on the diet, this is the 1 month of profiling, you can see that we virtually eliminate all spikes in glucose levels. And this is for the entire now half a year of the study for this one participant. Here's another trace of another participant. At the top left is the pre-intervention profiling period. You can see many spikes in glucose levels, and then most of them go away once we start the algorithm-based intervention. Here's another participant and another. And here's now looking at all participants on the algorithm diet. You can see the difference between the average response to meals in the pre-intervention period in red compared to the post during the intervention period in green. You can see clearly the differences at the individual level. And in contrast, when we look what happens to people on the Mediterranean standard of care diet, this is what happens. Normally we don't see this because we don't put CGMs on people. But if you were to put a CGM on somebody going on a Mediterranean diet, you can see that on the top left is the pre-intervention period, then you have the intervention period, and by eye it's quite hard to see that there's even an effect. Here's another participant. And when we look at the final primary outcome results of this clinical trial, we can see on the left here, hemoglobin A1C after 6 months in the middle goes down, but then after the 6 months of follow-up, it reverts back to baseline levels. In contrast, we were happy to see that on the algorithm diet, we get double the effect after 6 months, and that effect remains in the 6 months of follow-up. And to put this in a broader context, the NIH did a very pivotal study called the Diabetes Prevention Program study that I'm sure you're familiar with 3 different arms. And the algorithm diet was better than even the lifestyle arm, which included not just a dietary change, but also enhancement of physical activity and caloric restriction, both of which we did not do in this study. And even to put this in broader context, the NIH study showed that when you follow people for 5 years, the lifestyle arm reduced the conversion of pre-diabetics to diabetes by 60%. And since we got an even bigger effect, we're hopeful that the intervention we propose would reduce the conversion to diabetes by even further. So this was kind of a quick summary of about a decade of work going from idea to the first study on 1,000 people to then a proper randomized clinical trials, which took us on pre-diabetics, which took us several years to complete. And when we finished the study, which focused on microbiome and nutrition, we thought, more broadly about what we've done here was to look at, for the first time, maybe a large-scale, data-driven approach to the problem of nutrition, of personalized nutrition. And so we thought, can we take an even broader approach to the data-driven approach to look at disease more generally? And the idea is also based on, what we've learned in the past 20 plus years since we had the sequencing of the human genome. And if I take you back to the year 2000 when the human genome was published, we knew back then of only several variants for a few diseases. And 20 plus years later, now we know of hundreds, sometimes even thousands of different genetic variants for every single disease. So this was a major advance. But of course, genetics data is just what you're born with. It doesn't take into account your lifestyle, your nutrition, your exercise, and so on. And for that reason, people started looking at other layers of data, what is broadly called omics data. So people started looking at the microbiome, they looked at the immune system, RNA sequencing, they look at metabolites in blood, and so on and so forth. But these have also been typically narrowly studied, one omic levels at a time. And what we thought is, what would happen if we were to take all of these omic layers and measure all of them on the same people and do that for a large number of people? And the idea is that we believe that because much before clinical symptoms of disease appear, they would have some manifestation at some level of omic layer, then if you were to do such measurements on healthy people, you'd be able to detect the trajectories of disease well before they appear. So you'd be able to find novel disease biomarkers. In some cases, this early detection could lead to interventions and lead to new personalized treatments. And so on this premise, we started what we call now not the Human Genome Project, but now the Human Phenotype Project, to look more broadly than the genome. And I believe this is what is going to take us, I hope, to the next generation of precision medicine. So this is not just an idea, this is an actual project that we started in the lab over 4 years ago, the Human Phenotype Project. It has at the core the data, or what we call a library, because it's not just data, library has the connotation of knowledge. On top of it, we provide a platform that gives access to the data. I'll elaborate on that more because I think it may have relevance to people here, because we do give access to this very vast amount of data. And then many applications that could be developed on top of this data. We so far recruited over 9,000 people. We follow them longitudinally, so we have over 30,000 human years that have been followed in this project. And we do everything in my lab, starting from recruitment of patients, bringing them to the clinic, profiling them, collecting samples, processing samples, and then of course the core is the data analysis. So what is this actual data that we're collecting? So every participant that comes to the clinic at our institute undergoes all of this battery of tests that is shown here. So first, he or she provides their historical blood tests, their historical medical records. Then they undergo an ultrasound test to look at fat in the liver, to look at carotids, so to look at cardiovascular disease. With a DEXA machine, we look at full body composition and bone density. For the cardiovascular system, we look at EKG and ABI, which measures differences in blood pressure between the arm and the leg. We take high-resolution images of the retina, from which you can see many blood vessels. We connect people and send them home with CGMs, also with sleep monitors that for 3 nights monitor with several analog signals their sleep quality during the night, which, as far as I know, has never been done on thousands of people, definitely not coupled with all of this other data that we have. We know all the medication that people take. They log their diets in a log of 2 weeks while they're connected to the CGM. They fill out hundreds of medical background, lifestyle, and so on questionnaires, and we get all of their diagnoses. So this is kind of the clinical assessment. In terms of the omics, we do full human genetic sequencing, oral and gut microbiome. We do metabolomics, probably the most interesting test that we do, thousands of different metabolites that we measure in the blood. We look at proteomics, so proteins that get spilled into the bloodstream. We have a novel immune assay that we developed with hundreds of thousands of antigens that we synthesize and then look in one experiment by taking antibodies of a person. We know all of the antigens that are recognized by the antibodies of a person. So if you will, we can get the entire infectious history of a person. And we do RNA sequencing from blood to look at RNA levels. We also biobank blood cells and serum so that we can apply in the future additional tests. So this we measure for all people. We do this for a cohort of healthy people between the age of 40 to 70, but in addition, we also work with clinicians and establish these types of measurements also on several diseases, including cardiovascular disease, several different oncology cohorts, fatty liver disease, and atopic dermatitis. So this is the data. We also are developing a platform that enables access to the data, because it's very clear to us that we will never be able to extract all the discoveries that could be made from this data. So we are looking to provide access to the data, and we have a spin-off company who is providing that. So if this is of interest, feel free to contact me. And we believe this will have a lot of interest, because if you look at projects like the UK Biobank, which I'm sure you heard of, this is a very fascinating study of 500,000 people, but it started 15 years ago and has not the depth of data, definitely not omics data and other measures that we take here, yet it has been accessed by tens of thousands of researchers. So this complementary and unique data that we're developing, I think, will have a lot of interest. If you think about the nutritional angle here with the CGMs and the nutritional profile, to couple that to sleep quality and to all the other phenotypes and omics, I think creates a very unique data set. So in the last several minutes I want to tell you about a few discoveries that we started to make from this data, starting from therapeutic approaches. And here we've been starting to develop personalized probiotics, so probiotics for therapeutics. And we can do this because we have a very large microbiome data. And so here too, like we did with nutrition, we want to follow a data-driven approach to discover which bacteria we should give people in order to treat different indications. And the ones we started with are diabetes and weight loss. And the idea is that we developed a discovery platform that starts from the microbiome samples of people, but now we have one of perhaps the largest cohort of microbiome that's coupled to all this other data. And so we can look not just at the composition of bacteria, but really do what people do in genetic analysis, human genetic analysis, do this for bacteria, and look at genetic variation on individual bacteria at the single position level and see if that's correlated to indications that we care about, in this case like BMI. And when we do that, we actually have been discovering several strains of bacteria. So now this is the same species of bacteria that exist in people, but if you look at different strains of it, the blue strain and the green strain, we see that people harboring the blue strain have on average 2 points lower BMI than people harboring the green strain. And 2 points of BMI is obviously huge. It can be 10 or even 15 kilograms of body weight. And not only that, but when we see what happens to these people 2 years later, we see that people who harbored the blue strain did not gain weight, whereas people who harbored the green strain gained on average another 1 point of BMI, which again is a lot. So this, of course, does not prove causality, but it provides suggestive evidence, and eventually we need to go into clinical trials. We have been identifying numerous of these strains, and the idea now is to work on isolating them, and then hopefully building rationalized probiotics that we can then test in clinical trials for their efficacy, in this case for weight loss, and we have others for other indications like high blood pressure, cholesterol, and diabetes. Just to show you that in other work that we've done, bacterial interventions can actually have therapeutic benefit. So this is a little bit outside the scope, but nevertheless, for atopic dermatitis, we recently published work where we've taken patients where I hope you can see here clearly manifestation of this skin disease, where after we've given placebo, so nothing happened, but after we've given, in this case, this was a very coarse fecal microbiome transplant from healthy patients, we were able to eliminate the clinical manifestations of disease. And here's another patient, even many weeks after we've done the FMT. And on average, participants in this very small proof of principle study on nine people, 100% of them responded with an average response of 70%. And when we looked at the microbiome, we saw that here are the donors in ellipses and the participants, and after placebo, there's no change in their microbiome. But once we provide this FMT, participants really have a vast change in their microbiome, which becomes now not identical, but much more similar to the donor who donated samples to them. So we believe that this microbiome alteration was behind the therapeutic benefit. And here too, we are now working to isolate the relevant bugs and see if a probiotic or a formulation of the relevant bacteria could by itself be a therapeutic for atopic dermatitis. As another example, we can use this very large cohort to also find biomarkers of disease. So in this case, we looked at cardiovascular disease and have taken a novel precision medicine approach. So here, the idea is that even if you have a single acute coronary syndrome patient, so a patient who underwent a heart attack, you can go into our healthy cohort, find matched individuals for this patient. Let's say 60-year-old male, you find 60-year-old males in our cohort, and then you can compare the metabolite levels of this participant to the matched healthy controls. You can find all the metabolite deviations compared to a proper and relevant reference group for this patient. And then you can use all of the data to ask, what are these metabolite deviations arising from? Are they related to diet? Are they related to metabolites that are controlled by bacteria, genetics, or traditional risk factors? And when we do this, and this work was published in Nature Medicine, I'm obviously going through this very rapidly, we can see that even though we have different patients who are at the same age, 60-year-old males who underwent a heart attack, the underlying reason for that are different metabolite deviations. In one participant, they may be deviations in metabolites that are controlled by gut bacteria, whereas in another, they can be deviations controlled by diet, which suggests that the treatment for these patients should also be quite different. And finally, in closing, we have a large program in oncology, where we're doing also bacterial-derived interventions, some also just prospective studies, and also some dietary interventions. And I want to just mention this intervention that we're doing in breast cancer, because in breast cancer patients, over 75% of the women are hormone receptor positive, in which case they receive endocrine therapy, which is efficacious, but also has side effects of weight gain, which itself has many different complications. It also causes women to stop the treatment. So obviously this is something we want to target. And so here too, we've been carrying out a personalized nutrition study with two arms, a Mediterranean diet and our algorithm-based diet. And we've seen that not only are we able to eliminate the weight gain, but also actually we're achieving weight loss. In this case, we're achieving similar weight loss on the algorithm diet as we are on the Mediterranean diet. But when we connect these women to CGMs, we see that the algorithm diet is also having the benefit of balancing their blood sugar levels, which has many health benefits, of course. So in closing, I'll just say this entire project, we are interested in scaling it up on at least 4 different dimensions, scaling up the number of participants, the assays that we do, also the diseases that we look at, and also in terms of geographies that we'd like to establish and have partnerships now to go beyond profiling just the Israeli-based population. We do, as I mentioned, everything in my lab from building the cohort to analyzing the data, and we're open to collaborations to use our entire platform to profile different populations of interest. And I believe and hope that all of this line of work will really take us to the next generation of precision medicine. With that, I'll close and have the acknowledgement slide of the many people in my group who have been leading this study, and thank you for your attention.